So today, I'm going to take advantage of this seminar, this Krasnow seminar, for celebrating a special birthday, which is birthday number 10 of a project that was started and still thrives here at Krasnow, namely Neuromorpho.org, a database of digital reconstructions of neuron morphology. And along the way, I will try to, in passing, make a few comments on what works and what does not work in neuroscience with respect to data sharing. And I will also try, because we are Krasnow, to connect the shape of neurons. And what we're trying to do is quantify as much as possible the data of these neurons with the mind, and why I actually think that studying the arbor shape of brain cells is absolutely essential to understand how the mind works. So we'll start from there, and this is because it's Halloween, I figure I should put a postmortem brain picture. So this is a human brain fast frozen after death. It's reverse contrast, so what you actually see, this is mildly stained. What you see that is very dark and black is actually the white matter, and what you see in brown or purple is gray matter. And if you were to take a similar brain and stain a different way, so this is not the same specimen, but it's tissue taken from the University of California, Davis brain bank, Nissl stain, you'd see cell bodies of neurons, and these are various frames from various parts of the human neocortex, that look basically like normal cells, like the kind of cells that you see in textbooks, like kidney cell or liver cells or blood cells and so forth. If you squint or if you close pay attention, you can actually see the nuclei within the cytoplasm and the membrane. But what is very interesting about the neurons is that if you stain them the right way and you tune the magnification the right way, you'll see that attached to the cell body there are arbors. And in fact, the arbors are so massive relative to the cell bodies that completely dwarf the size of the cell body. So the cell body, which in neuroscience we call the soma, is basically the same size roughly as other biological cells in other organs. But the arbors that come out of the soma are both in terms of span, 100 or thousands of times bigger the size, but also in terms of surface and volume that constitute 99 or 99.9% of the actual neuronal physical existence. And the other very interesting thing is that although all the cell bodies look more or less the same, if you look around the brain in different brain regions or in different developmental stages or in different animal species, or even within the same brain region of the same animal in the same developmental stage at any one time, you'll actually see that the trees come in an incredible variety and diversity of shapes, very much like botanical trees. So if you walk around in Berkeley Park or in any other nice Fairfax Park, you'll see that there are trees of very many different shapes, and it's the same for neurons. Now, it's true in nature, no two objects are ever the same. It's true for the neurons, it's true for botanical trees, but there are also different kinds of trees, right? So you can find, say, two oak trees. They're different, but they're still oak trees, or you can compare an oak tree to a pine tree, and they're definitely more different. And the same is true for neuronal trees. There are different cell classes as represented here, or within a class you find differences that are quantitatively there, but less qualitatively obvious. The other striking thing is the extent of these trees. So I'll talk about axons and dendrites. This picture is about axons. And if you now take, this is actually part of the image, you see pixelated here because it's blown up. So these are cortical folds of this human brain. And these are basically branches virtually embedded. So this is artistic rendering, if you like, but it's in scale. So these will be filaments of axons that are coming from somewhere, probably in the striatum, since the ram is here, and they're projecting somewhere in the cortex, and different colors for different neurons. So the arbors and the branches, the branching structures of these neurons can actually extend through macroscopic spans in the brain. So from ear to ear, or as you can see when you think about moving the pinky toe in your foot, there is basically one tree that goes from the top of your skull all the way to the bottom of your spinal cord, and yet another tree that goes from the bottom of the spinal cord to the muscle controlling your toe. So it's only basically the span of two neurons that are covering the whole length of your body. Right? If you're a basketball player, it's even longer. So I'm going to, having introduced the players of this, I'm going to give a very quick perspective about the numbers, because the entire talk and much of the operation in my lab has to do with individual neurons, but it's very important to keep in perspective how these neurons work together in a given brain. And the key thing about the brain and about these arbors is that it's a network. So the fundamental element there is called a synapse, or a connection between a sending neuron and a receiving neuron. And every neuron works as sender for tens of thousands of other neurons, and it works as receiver for tens of thousands of other neurons. So the synapse is basically a contact between one axon, which is the output, I'll explain this, and the dendrite, which is the input. The output typically of one neuron and the dendrite of another neuron. And the number estimates for the human brain is that they're just shy of 100 billion neurons in a typical adult human brain. And on average of the order of 1,000 trillion connections. Okay? So this is a very large number. Each of those neurons have these trees, but they're a very large number in the brain. And this is, as an order of magnitude, roughly the same as number of real trees. And by real trees, I mean the ones that have a stem of the size of a human body. Technically speaking, the definition of a tree is much more permissive, so there are actually many more trees on Earth than this. But if you consider the trees as kids imagine them, with the trunk and limbs and so forth, then it's about the same order of magnitude. And roughly the number of leaves per tree is also of the orders of 10,000 or maybe a little more. So you can imagine that the number of synapses in your brain would be roughly equivalent to the number of leaves on Earth at any one time. And here is a trick. This is how we are able to pack all these trees in the brain, is that the branch thickness is actually much, much smaller than you'd expect at scale. Okay, so in reality, it's about a tenth of a micrometer or so. A typical human hair is about a tenth of a millimeter, so it's 1,000 times thinner than that. But the length of the neuron, if you sum all the branches, is actually macroscopic, right? So you measure it in inches or even feet if you consider the axons. So that means that if you were to take one of these little neurons in your brain and you were to blow it to the size of a regular oak tree, let's say, the trunk itself would be about the size of one of your fingers. And the little side branches would be about like spaghetti. Okay? So very, very thin proportionally to the span of the tree. Okay? And the fact that the branches are so thin allows real brains to pack an incredible number of these trees. So you have 10 to the 11 neurons. Each of the neurons can have an actual length of, say, a few inches. When you sum it up, it turns out that the total cabling in a human brain is literally hundreds of millions of miles. And this is not scale. I'm not scaling it up to a real tree. Okay? This is the actual nominal length of all the axons in a brain. And again, just for perspective here, this is, according to Google Maps, just about the length of all the roads, the paved roads in the world combined. And it's also about 5,000 times the circling of the equator. This is just in a single brain. So I've been doing a whole lot of flying for conferences and so forth, and so I take the luxury of counting that it would take 15 years full-time of a jet airliner with a pencil attached to it to draw that line and trace all the axons in a single human brain. Okay? So the numbers are actually quite impressive. And now I'm going to give you a kind of a crash course of 3 minutes of Neuroscience 101 and defining what dendrites and axons are, because if I'm not allowed to talk about this, it will be very hard to explain why I'm so enamored of these trees. But basically, the axons are the outputs of the neurons that carry signals from the presynaptic neurons to their target. And so this is a picture of 4 different axons of a particular neuron type in the hippocampus, and they're color-coded. Whereas the dendrites are the input, so they integrate the signal from many sources into the postsynaptic target. Okay? And the key thing about the scale is that the axons are much larger, span a much larger size than dendrites. So in scale, that one dendrite here, which is of the same type of neurons, will be about this size. So the axons are much bigger than the dendrite. Does this make sense? I'm probably saying something that you guys all know already, but I figured I would say it anyway. So the axons cover most of the distance, but the dendrites do most of the computation. And specifically, the dendrites are responsible for the large amount of plastic changes in neurons. So a lot of what changes in neurons as you learn things and so forth occurs in dendrites. And lastly, they communicate in different ways. The axons use basically binary codes of spikes. Now, everything in biology, as soon as you say it, you have to mention exceptions. And there are many reasons to think that this is not exactly true. But with first approximation, it's actually a very good way of thinking that basically axons work in terms of zeros and ones. The ones are the spikes, and zeros are lacks of spikes, so similar to the code of digital computers. Whereas dendrites basically can use spikes if they want, but they can also use analogic and linear code or proportional, so they basically can do anything from 0 all the way to full-blown spikes. Okay, so they are very different beasts, they do different things, very complementary. And with that in mind, I have now for several years maintained that the branching structures of the axons and dendrites, of course, determines where the synapses are, because the dendrites are formed largely from the axons of the presynaptic neuron to the dendrite of the postsynaptic neuron. But I'll try to convince you that this actually defines the space of what can be learned. Okay, so this is actually where the connection in my mind, at least, is between brain activity and mental content. Okay. So this is basically my intro. I'm done with my semester worth of course, so from now on, it's mostly philosophy. So if you have any questions, this is a great time to ask. Yes?
So could you elaborate a little bit more about defining the space of what can be learned?
Yes, I'm going to elaborate in the next 3 slides. So, here.
I'm actually a plant here.
Yes, this is perfect. Now, before we jump into the theory, I want to say that just based on this fact that dendrites and axons are known to do a lot of what the brain does, except at the cellular level, there is a whole lot of experimental and empirical science out there of people trying to understand and characterize these neurons and what they do, and try to get some gleaning of information about what the function could be, but also what goes wrong during disease or other conditions. So people ask these questions about the morphology of the neurons, the structure of these trees, and they say, is there a difference between, say, the excitatory and inhibitory, the trees that carry out excitation or inhibition? Is there a difference in when neurons develop and mature? Is there a difference between the trees of developing neurons or mature neurons, or young and aged individuals, or across species, say, between rats and humans, if we look at the same brain region and same neuron type? Or within different brain regions, say, between superficial neurons and deep neurons, or in healthy controls and Alzheimer's patients, and so forth, genetic manipulations in animals, dendrites before and after experimental models of potentiation and plasticity, or even after a lifelong intake of heavy caffeine, which would describe very well my own brain, could you see a difference there? And of course, these are actually very challenging to do, because if you're looking at a mouse, say, you cannot, in the same individual, both give it a lot of caffeine and not, so you have to do it in very careful ways in order to control it statistically. But if you open a random issue of Neuroscience or even Nature Neuroscience, occasionally they make them all the way out to Science and Nature and so forth, you'll see a whole lot of data that has to do with, oh, look, when this happens, the structure changes, or we see a branch that goes up or comes in, or so forth. And so there is a lot of evidence out there. And before I jump into what we're doing with this, which is basically trying to put it all in a common public database for reminding, I'm going to spend a few minutes on theory. And this is actually an example from many years ago. And this was one of Harold Morowitz's favorite studies, because it was in collaboration with Jeff Kretschmar, who's now the collaborator at University of California, Irvine. but basically puts together the morphology, the molecular phenotyping, and the electrophysiology. And this is data that came from Stanford University, from Rob Malenka's lab. And these are images of cultured neurons in control conditions with an overexpression of this growth factor, beta-catenin, and the genetic manipulation that basically removes much of the endogenous beta-catenin. And in my lab, we reconstructed these neurons. So these are digital reconstructions of these neurons extracted from Malenka's image stacks. And then Jeff Kritschmar ran simulations where you could basically separate the variables and say, we're going to put exactly the same mathematical equations describing the electrical activity in these neurons, and the only thing that is changing is the actual morphology. And we obtained results that are striking difference. And if you've ever done or read or seen electrophysiology, you realize that these patterns of firing are basically the gamut of how different neuronal firing can possibly be. And these were basically taken with the exact same mathematical equations, the same electrical properties, except the morphology of these neurons is different. Okay? So basically, you have these connections between, in this case, molecular effectors that change the morphology of the neurons and the effect of that on the computation. So there's a lot of interesting science that can be done if you are just willing to play with these data. So I promised I would try to justify why I think that this is actually relevant to cognitive science. And so I'll shamelessly introduce what I consider principles of brain-mind relations. Some of these are actually very trivial, and I think that most people in their own right state of mind would agree with those, except that they are very rarely explicitly stated, and I think that neuroscience would do better in explicitly stating these. So the first one is that mental state are spatiotemporal brain patterns of electric activity. This is a very general statement. I don't go into the details of what that means. I don't go into the many different possible theories of how that would be instantiated. But basically, this means that when we think or when we have emotions or when we have any mental state, including meditation or dreaming or pretty much anything that we are alert of, that corresponds, or in fact is, spikes distributed throughout neurons in your brain. You can disagree with that. I actually once ran a poll at Society of Neuroscience and found almost everyone would agree with that statement, with a few exceptions that I'd be happy to discuss over beers later. So after having told you how beautiful dendrites and axons are, this is my cartoon characterization. This is a cell body, a dendrite, that's a little spine, so the input. This is the axon in red with the little circle that corresponds to the output. And so this will be a synapse between the horizontal neuron serving as presynaptic and the vertical neuron serving as postsynaptic. And that will be a whole network, in this case of 12 neurons. You'll notice that there are two kinds of neurons, the black, which I'm envisioning to be excitatory, and the gray that I call inhibitory. But really, it's just a cartoon representation of a very small network of neurons. And then I created this little script that propagates activity with some very silly equation from the presynaptic neurons and the postsynaptic neurons. So this will be a pattern of electric activity in a very cartoon representation, if you like. Okay? So in this little very stupid animal with only 12 neurons, this will correspond to a mental state. Now, if you think that mental states are activities, because the patterns of activities propagate through synapses, very much like traffic of cars propagates through the grid of roads, if you want to think about all possible mental states that a brain can have or an individual can have, which basically is the body of knowledge and personality that that person has. So if you want to basically try to imagine the entirety of the mental repertoire of an individual, the best approximation, at least that I can come up with, is in fact the connectivity of its brain, because the patterns of activity have to flow through the connectivity of that mesh. Okay? And this is, in fact, again, I try to express it in words and make it very explicitly, but this is my understanding of the cognitive and philosophical justification about the rave about connectomics as to why people now are after trying to, not sequence, but reconstruct the connectivity of entire pieces of the cortex in mammals or the entire fly brain in its entirety. And basically because ideally, by reading the connectivity of that network, we should be able to understand what its knowledge is, because we will be able to encapsulate all possible individual patterns of activity from that. Now, of course, the connectivity itself is not sufficient, because we also need to know what the input-output relationship of those neurons are and the weights of those connections, etc. But the connectivity is, by almost certainty, necessary for that. And please feel free to interrupt any time. Worst case scenario, we don't get to the end, but that's also fine. So that's kind of what I call the first principle in green, and this is actually a corollary, if you like. And now, when you think about what learning is, well, when you learn something, let's say I learned a fact today I did not know yesterday, and tomorrow I can either recite that fact or simply think of it as a piece of knowledge in my head that I did not have the day before. So I've changed my knowledge by learning something. And so if the knowledge is basically equivalent to the whole connectivity of the network, it goes without too much of a stretch that learning would correspond to changing the connectivity of the network. And indeed, although the first principle, that first principle up there, is widely agreed upon in the community, this second principle is not completely cuckoo, but I would say it's a hefty minority of the neuroscientists out there who think that knowledge, in fact, that learning, in fact, as alteration of knowledge, is change of connectivity. So this is kind of the theory of structural plasticity, where you say that when you learn something, you're changing the synapses in your brain. You're literally taking out some synapses, adding some other synapses, and so forth. And there is now increasing experimental evidence supporting this, but it remains a theory until it's proven in a definitive way. So
Yes. An only if or just if?
No, it's an if. Well, it's an if, and not only that, but there is obviously a competing theory. This is one of the reasons that there is so much resistance, is that in the world of especially theoretical physics, the classic theory is that it's the weight of synapses that correspond to, an alteration of the weight of synapses correspond to learning. And I'll show you in my summary in a few slides what I think that actually is. But I would say most people at this point, even those who believe that connectivity is essential for learning, that basically what we go on is a combination of both changing connectivity and changing the weight of existing connections.
And no notions about growing new neurons?
Yes, there is absolutely growth of good neurons. So in fact, there are different temporal scales that I'm not going to go into because it would take quite a while, but there is definitely growth of new neurons in specific parts of the brain, especially those responsible for episodic learning and memory, which is, of course, very suggestive. But also there is a gross alteration of the morphology as well over long time scales. And by long time scales, I mean weeks, months, years. The change of synapses, experimental evidence goes, can occur really, really quick in the span of a few seconds or half a minute, which is typically what might take an individual to learn something new.
So I was trying to understand whether or not it's controversial because it's not very specific, or it's interpreted as being too general. I mean, there are lots of different kinds of learning, and there are lots of different changes that one can imagine to the brain.
Great question.
And so it seems like,
yeah.
Depending on which hat you're wearing.
I think that the reason it's controversial is because people are hanging on the idea of weights, and the early evidence was that synapses don't really change. And part of the reasons for that is that the resolution of the experimental technique was not sufficient, but also because it's actually rare events for synapses to change. And in fact, when you think about, even when you learn something, you actually change a very small, very, very, very small amount of the body of your knowledge. So to catch that in the act, in a behaving animal, it's extremely difficult. I think that's my opinion as to why this is still controversial. But I mean, when people started, just a second, when people started, like maybe 15 years ago, putting this out, it was, basically a few crazy people. And now I would say we're close to 50% of people believing that. And there are like major funding efforts that are based on this assumption. Yes, Aram, sorry.
So, I'm going to say you asked me a similar question about what about growth of new synapses. I don't see why you're making such a strong distinction, because in my mind, a synapse with a weight of, and I'm normalizing this, 0.01 is the same as no synapse.
Yes.
So I don't distinguish those two.
Yeah.
It has a range from 0 to some And zero, going from 0 to 0.01, say, is creation of a new one, and 0.01, the rest of the range is growth. Why are you making the distinction?
It's a great question. I think that the minimum is in fact 0, but there are cases in which it is going to be physically impossible for a synapse to be there. So I think that a synapse, so you can imagine, if you like, a potential synapse or an overlap between an axon and dendrite as a synapse with 0 weight. Now, there are biochemical reasons that this is not the case, and you have both states. You have the synapse not being present, and the contact with the shaft being present, and yet the weight is 0. Okay, there is no neurotransmission there yet. The reason I'm making the distinction, I'm saying actually presence or absence of synapses, is that in certain situations, I will argue in my probably next slide, synapses cannot possibly be formed.
Right. So in my mind, that distinction is only important for those very abstract networks where they would, think that you could associate two different synapses. But if you were, let's say, you're creating a more realistic network where you don't say have built in this absolute, detail exactly what the connectivity is, but you have things like a topographic map.
Absolutely.
Then the distinction is actually irrelevant, I would claim.
To this theory it is. I think there are biological reasons that the distinction is not irrelevant, but it is to this particular theory. But keep in mind that most neural network theories are in fact of the abstract kind, so they don't look at physical embedding. So, I mean,
But I could give you a whole host of other reasons why those studies are
Sure. I don't, since we agree on that point, I don't think we need to argue as to what the motivation is as to why those studies are wrong. Okay, so because I had shown the pattern of activity, in this case, I'm hypothesizing that these two synapses get formed, and I'm using here very simple Hebbian dynamics where neurons that fire together, they wire together. So in this particular case, the presynaptic neurons is causing depolarization in the postsynaptic neuron, but the postsynaptic neuron was already firing, so new synapses are formed. So according to this theory, creating synapses requires an experience, and this can be that you live an episode, somebody tells you something, but it can be actually much milder than that because we're talking about mental experiences, so you can simply sit on your chair with your eyes closed and think intensely about something, and eventually you say, aha, and maybe you solve the theorem in your mind. You haven't said anybody, you haven't written it down at all, and yet clearly you have a new mental state. It's an important one. You are looking for that answer, and even if you get distracted or if you fall asleep, there is no way the next morning you would have forgotten that you found that solution. Okay, but these are basically one-time, very fast connections that last in the scale between seconds and minutes. And again, there are many kinds of learnings. There is also repetition learning. There is emotional learning. I'm talking about the most neutral possible, one-time, non-emotional learning. And here is where I think that axons and dendrites become very, very important. And I'm going to try to define what I said before, which is that their shape basically define the space of what we can or cannot learn. Is that in the vast majority of cases, the same experience would not trigger the formation of new mental states. So in other words, we actually learn a very small fraction of everything that we could learn, okay? Or we remember a very small fraction of the episodes that we lived. As a matter of fact, that's actually a very nice trait of individuality, meaning that if you take two people, even from the same background, speaking the same language, even siblings and so forth, in the same context, or attending the same lecture or watching the same movie, and then you ask them a test, they will have learned different things and they'll remember different details from that. Okay, but most importantly, the key thing here I'm trying to convey is that in most cases we don't remember stuff that we experience. So it seems that more than just activity would be needed to form new synapses. And I mentioned this is philosophy, so bear with me. It's not. I don't have much experimental evidence in this case, but these are examples, and I'm trying to drive the point that somehow background information is important. It's a good discriminant. In other words, if you know something about a subject, it's much easier for you to learn new things about that subject, and this is well known in psychology. So, for example, a Mason student noticing once that the Q bus now stops in front of the church, they use this information next week saying, oh, I'm going to pick pick it up there since it's closer to my office. Whereas an out-of-town visitor, first time on campus, sees the very same bus stop in the same place, but will not be able to remember it a week later. So this is an example of a piano player, so proficient piano player, just glancing at music scores will learn to play the piece or singing it or humming in their own mind, whereas beginning players, they can read the notes just as well, but they will not be able to learn it or not even imagine how to play it. And this is for my wife, who's a mathematician, and she might read a new demonstration of the proof that the square root of 2 is not a rational number, and she will absolutely remember it. And even if you know what it means or can have a scientific American level, if you don't have the required basic mathematical skills, you'll probably forget that demonstration in a few minutes. And of course, this is for soccer because I really like soccer. And if you are into soccer and you look at the score, you immediately realize the consequences of that in terms of the qualifiers for the World Cup, etc. And if you don't know those background essential pieces of information, you might forget, and so forth. So the theory of this is actually something that I put forward a few years ago. I actually first tried to explain this at a Krasnow seminar many years ago. But it's what I call a basic principle, but a new principle in the sense that it's neither a majority nor a minority. It's just my own speculation here. That basically the overlap between axons and dendrites is the neural correlate of the capability to learn. And this basically starts from something that is very obvious, and it's so obvious that it's not a matter of opinion. It's very clear, which is that new synapses can only form between the pairs within a pair of neurons if the axon of the presynaptic neuron is close physically to the dendrite of the postsynaptic neuron. If the axon is here of one neuron, the dendrite is there, you can fire together all you want, but you're not going to wire together. You're physically impossible. You have a physical impossibility to do so. And this is true for any plasticity rule, STDP, LTP, whatever. It doesn't matter. You simply can't. And the reason that I think that this overlap between axons and dendrites is in fact related to background information gating, or the expertise requirement for learning, has to do with a moment of, I guess, serendipity that I had several years ago sitting on my porch, and I realized that there was a buzzing beetle flying by, and I simply did not know, city boy that I am, that beetles can buzz. And I honestly cannot care less. It was the first and probably only time I saw a buzzing beetle. It was not emotional. I did not get stung. I was not scared or anything like that. But it's certainly something that obviously still remember. And so in my mind, if I can borrow the cartoon of grandmother neurons, so pretending there is one neuron that represents the concept of buzzing in the brain, and a cartoon of a neuron representing the concept of beetle, I figure that I'm now associating those two, and that means that there must be some reason that the buzzing axon was somewhere close to the dendrites of beetle. And the question is why? Because you see, we are, yes, packing the arbors of 100 billion neurons in the brain, but the space is not infinite, and we're still space limited. Okay, the reason that we have 9 months gestations is in fact that there is only a certain amount that skull of babies can be given birth to. Yes, Abraham.
I think it's to allow the mother to get time used to the idea of being.
All right, that sounds good too.
Do it personally.
Social evolution, yes.
So I'm not exactly sure where this would fit in the scheme of what you've just been talking about. But if you take a neonatal hamster and remove the superior colliculus shortly after birth, before retinal collicular connections occur, then those axons from the ganglion cells will grow into the inferior colliculus, which ordinarily would be auditory. And it would seem that, I don't know if that's a violation of the rule or a different way of interpreting things, because...
Not at all. It isn't. Let me say it's very clear, but it doesn't happen in a takes many days and weeks to reroute. It's definitely structural plasticity at a much longer time scale. And the truth is, the same is true, in fact, for normal course of development. Neurons migrate, they form connections. It's quite clear that babies learn languages, and the very same baby, if you grow in Japan, you learn Japanese. If you grow up in Italy, you learn Italian, and so forth. But those things take time. It's not an instantaneous one-time learning. Okay? So I think that in general, you can have very broad rearrangement of these entire trees given enough time. But at the level of a few seconds, if you have an axon coming in a fiber and then right here, you can make that synapse in basically a few seconds. I mean, the measurements that people are looking at depends on proximity. They vary from a few seconds to maybe half a minute. But they're fairly liquid. The membrane is fairly liquid. Okay?
Yeah, I guess it would be new synapses can only form nearly instantaneously.
That's right. So this is, I kind of qualified it in the previous slide, which is this is emotionally neutral one-time learning. So I'm not pouring a huge amount of neuromodulatory chemicals in there, and I'm not waiting half an hour. I tell you something, and I test you tomorrow if you remember it, or in a week from now. So now the question is, if I learn that beetles can buzz, why would that be possible? Because if you take any two pairs of random neurons, the chance that they will have an overlap is basically nil, statistically speaking. Right? So you have 10 to the 15 neurons. A neuron only makes 10 to the 4 synapses. So the chance that you would be close to another neuron, just if it were random, it's basically very, very small. And so the reason is that I think that the axon is there for a reason. Again, they're kind of an optimization of cabling and location. So if the axon is buzzing is there near beetles because it's making connection to something nearby. And so the first question that we can ask is that what do we know that buzzes? And let's say wasp. I know that wasps buzz, and many other, in fact, creatures and insects. And so we would think that if beetle is there, it might be that beetle is close to wasp in the brain. And in fact, there is good evidence for topographic organization. So there are now, with functional imaging, typically areas are found that code for specific classes of concepts, such as insects, as a matter of fact. And so now the next question is, why would beetles and buzz be there? And the reason is exactly the same. There might be a bunch of axons that connect to both. And so in order to minimize the cable, you put them nearby. So basically one branch can form both synapses, right? For example, what we know is in common between all these annoying insects that might or might not sting us, and one of them is that they fly erratically, for example. Okay. Small, possibly scary, and so forth. So basically by putting together this notion, you might actually, by pressure of cable minimization, figure out that buzzing would have to be near beetle. And this is very different, to drive Abrama's point home, very, very different from artificial neural networks. Okay, so this is absolutely, it has something to do, much more to do with microscopy, if you like, than with machine learning. Okay, so here is where I spent a couple of minutes to do the math, because we're able to do that. So this is my cartoon representation of my flight, wasp, buzz, and beetle. So the same players, but they're drawn that way. And I'm basically making my point with a little equation where I'm saying, well, is the concept of buzzing, does this, consider as an axon, does this have an overlap with the concept of beetle, considered as a dendrite? And so I'm saying that this might be true, or becomes more and more likely. The more concepts there are that we know are connected to this, such that you have to have yet another concept that is connected to both. So this is a mouthful because it's a two-step reason, right? So in order for buzzing to have an overlap with beetle, we both need a bunch of things that we know already are buzzing, and those things must be having something in common with beetle. The more in common they have, the more likely they are to be close. Okay, so I'm basically repeated in a more abstract form what I said before, but the nice thing is that this can be very, very easily expressed as an equation. So you can basically say for any, in this case, concept C and concept D, concept A and concept B, we can basically sum over all C's and D's. So over the entire network, we just look at all the other neurons or all the other concepts in the brain. And we'll say that the overlap between A and B becomes more and more likely, or if you like, is proportional to the sum over all C's and D's, such that A is connected to C, D is connected to C, and D is connected to B. Okay? And all of those three connections have to be in place. So basically, this is a binary function, which is 0 if the connections are not there, and you don't sum it. And in order to be summed, all three have to be there. And the nice thing about this is that if you remember your linear algebra, you can express this very easily with the matrix formulation. So if now you create a matrix that is basically a connectivity matrix, where all your presynaptic neurons are rows, all your postsynaptic neurons are columns, and 0 means that 2 neurons are not connected, and 1 means it is connected. So these are basically, each of these entries is one of these omegas here. Then basically, this entire sum, you can express it for all the possible overlaps over all the neurons in one sweep with a very simple product of the entire connectivity matrix times its transpose, which is basically what you do if you flip it over the diagonal, time itself. And this is a very, very cute and elegant way to express it, which also makes it incredibly fast to implement in MATLAB. Okay? It's basically a split-second operation. And so based on this, we're able to run medium and relatively large-scale simulations and basically created neural networks that, instead of being abstract neural networks, could actually obey this idea that you could only form new synapses if you were in agreement with the rest of the connectivity network, without having to simulate the actual dendrites and axons, which would take forever. Okay, so very, very quick mathematical shortcut, of course, at the cost that you have to believe in wiring minimization. So you have to believe that the reason that this neuron could connect to that is that these other neurons are in the right connectivity location. And so I'm going to go maybe a little quicker here because these results are published and are very clear. But what we did is we took from Wikipedia a bunch of pairing between adjectives and nouns, pretended that was our cognitive world. And then we took 2 categories in particular, all the nouns that are used to describe animals in Wikipedia and all the nouns that are used to describe household objects in Wikipedia. Again, you can find these categories just by searching around. And so this is a bipartite graph, and some adjectives can describe both, and some typically describe one or the other. These are edges that are basically probabilities of finding those 2 pairs of words together. And what we did is we created networks artificially that would be more expert in animals or more experts in household objects, right? So we basically created networks with personalities. And what we did is, for example, we taught a network 50% of all the connections about animals. So this was a uber animal expert who knows 50% of everything there is to know about animals, but only 5% about the household objects. So this is a person who would not really do well at IKEA. And then we found we created networks that basically do the opposite. They're uber experts in household objects and quite ignorant in terms of animals. And then we say, can you learn more associations just by looking at pairings of names? And lo and behold, the more specialized the network is, the more clear the result is. But basically, the experts in animals learn animal things much better. The experts in objects learn object things a lot better. Okay, and this is basically confirming what, in a way, our theory predicted, that we built the network that way. And this is all based on these hideous grandmother neurons, which nobody really believes in, because we know that concepts in the brain are represented by cell assemblies, not by individual neurons. And so we repeated this work with a network where the representation was distributed across many neurons. So we basically created a graph-like structure, and we tried to repeat the same experiment where now concepts are represented by cell assemblies, the way that modern networks use, and indeed reproduce the very same result, that expert learning is way better than novice learning. But then we found out something that is very interesting and we did not expect, but then we were able to explain, which is if you present 2 pairs of concepts at the same time, for example, buzzing and beetle, and kumquat and sour sweet, and we started doing this to speed up the simulations, to teach the network more things at once. incredibly, the network learns very well that kumquats are sour sweet and that beetles can buzz, but they do not learn that beetles are sour sweet and that kumquat can buzz, which of course is right, but it's a completely non-trivial result. Yes, Ernie?
How do you implement that experience?
We present at the same time. This is straight Hebbian plasticity, so wire together, fire together, wire together. And it's basically presenting 1, 2, 3, 4 at the same time. So you have 2 correct associations, and all the rest combinations are all incorrect. Okay? And then you basically see what is it that the network learns. And of course, you know what the ground truth is. You know what is real. And so what you find is that learning real things that are out there in the graph of reality is much easier than learning spurious things that just happen to occur together by chance. And again, this is one-time learning. It doesn't take next day to see again a buzzing beetle without tasting a sour sweet kumquat at the same time, right? Which is how classic theories have it. It's a one-shot. But basically, explanation is very much the same. So I just wanted to explain that there are simulations, there are theoretical frameworks that allow us to think of the branching structures of arbors as defining geometrically a very large state of cognitive space in which our minds navigate. So I think there is a very interesting, fascinating relationship between the geometry characterizing connectivity in the brain and the geometry characterizing connections in the mind. Yes, Kevin?
So, Giorgio, can this idea consider another proof for the small world network organization?
Yeah, I don't have the specifics, but yes, indeed, this is related to a small world network. I don't think they can be considered proofs, but certainly they use the fact that neural networks are small world. But basically, what the key parameter, I didn't put the slide in because it got technical, but the key parameter to allow these kinds of performance is the clustering coefficient. So the graph has to be at relatively high clustering coefficient, which again is quite well known in real biological networks. Okay, so I'm going to summarize this theoretical part because I do want to talk about neuromorpho.org for at least 10 minutes. This is a view from my office, a picture that was taken several years ago when the new wing was being constructed. And we're almost at the right season, maybe a few more weeks, and we'll get this similar view from our windows. And I find it as very inspiring because I like the winter, not for the freezing cold, but because the branches of the trees really work very well as whiteboards to think about dendrites and axons and so forth. So the summary of this is that I like to think about correlates between neural substrates and cognitive activity, if you like, or cognitive content. And so activity patterns in cell assemblies will correspond to mental state. This is kind of the first principle, and again, I think that it's relatively well accepted overall. Now, I think that synaptic strength, I didn't really talk about this, but I think it's much closer to the likelihood that we would think of something than the actual learning. So it's more like a matter of probability of having one thought instead of another, rather than the possibility of having that thought. And I think if the synapses are there, you can have that thought, but if the synapses are not very strong, then it might take better circumstances to have that thought. However, synaptic connectivity itself would correspond to knowledge learned from the past. And I emphasize that because in a way, the proximity between axons and neurons correspond to the potential to learn from the future. Okay, these are experiences you haven't had yet, but where you to have these experiences, you learn those facts. Whereas if you don't have an overlap between axons and dendrites, you can have those experiences, you would not learn them. Okay? And this is absolutely, when you try to summarize the brain-mind relationship on which thousands of philosophical tomes have been written over the centuries in a table of 5 by 2. Clearly, it's a gross oversimplification, so don't hold me up too literally about it. But in terms of bullets, is the way I like to think about brain and mind. Okay, so back to those trees. This is now, I hope I gave you at least a flavor of why I really, really like to think about these trees, both inputs and outputs, and why I think it's really important to study them quantitatively in all possible brain regions, in all developmental stages, in all animals, all conditions, both physiological and pathological. And the way that this is done experimentally is now literally almost every lab has different recipes. It's It's a little bit like your pumpkin pie recipe. You know, everyone does it in a slightly different way. But the steps are always the same. There is a step of labeling. So you kind of have to basically color these neurons somehow. Then you have to visualize them. So typically use microscopes and try to image those colors somehow. And in terms of labeling, there are many, many possibilities. Bulk loading, which includes the very, very famous Golgi staining, and now there are other bulk loading techniques that work even better than Golgi. Intracellular, that was very popular for about 20 years and continues to be used in quite many few different labs. Immunolabeling, that basically uses dyes or dyeing reactions attached to antibodies, and now the kind of all the rave popular technology, which is genetic manipulations, where you can basically insert fluorescent proteins directly into the genome of the animal, so you don't need to do anything, you just wait for the animal to grow up. And literally, some of the neurons are going to be green, blue, yellow, red, and so forth. Microscopy also is undergoing a spring. We had one of the Nobel Prize winners a couple of years ago here at Krasnow, Eric Batzig, who invented or co-invented one of the flavors of super resolution microscopy, kind of bright field microscopy, which is based on regular light and confocal microscopy, which is based on beamed light, are still the two workhorses of this, but more and more neurons are now visualized with electron microscopy, with array tomography, and a variety of other techniques, including light sheet microscopy and so forth. Then once you are visualizing these, then you need to trace this. So in other words, somebody has to take these images and create branching structures. And the tracing can be done, typically, historically, was done with a pencil on a paper. So you basically extract that signal out of the noise in an image. And it has undergone, over the years, a continuous computer involvement, more and more computerization, if you like. And now the idea is to bring it down to full automation, which is not quite there yet, but I think we're very, very close. I think we're going to get to automated neural tracing before we're seeing self-driving cars, but I think that the order of magnitude of the temporal dynamics there are similar. And finally, you get these digital reconstructions where the structure of a neuron is basically described as a bunch of XYZ coordinates and connectivity. So very much like you have a crystal structure of a protein. And in the end, that's kind of the bottom line of a bunch of science of data acquisition. You get numbers out, right? So any one neuron reconstructed will be a large file with maybe thousands of these rows that correspond to that structure. This is the data that we are aiming to store in neuromorpho.org. So we basically create a database where we say the distilled data of these reconstructions, we want it to be shared as much as possible. And so we started this project in 2006. These are some of the slides that I had made back then, and I still love to use them because of their historical values. So this is from version 1.0. Basically, you can. It's completely open access. You can download stuff. You can browse. You can do pretty much anything. You can search by metadata categories. You can display summaries and browse by a variety of dimensions. And I just came back from a short course in bioinformatics in Bogota, Colombia, where I gave a basically hour-long demo. And there were 2 hours questions of things. Can you search for this? Can you do that? And the answer was always yes, yes, yes. And it was a lot of fun to try to do that. But it's a very deep site. If you browse around, you can basically spend many hours on it and download a lot of data. This is actually version 1.1. But the key thing here is that in these 10 years, we've been accumulating a lot of data, and we constantly request data from the community and add it here. And I'm not going to walk you through the what's under the hood of the team here, but all to say that there are lots of steps in making sure these data becomes public. From the social engineering of actually requesting the data, which is still quite awkward in neuroscience, to, of course, processing the data because the authors are entrusting you with their data, and there is a whole relationship between metadata and the data themselves, and finally then the release. And all of that while maintaining a literature database that is kind of its own layer of metadata, because every single piece of data that is published in the database is linked to a peer-reviewed article. So there's quite a bit of interesting informatics there. The day before yesterday, we had a party at my house where we actually celebrated the birthday of neuromorpho.org, the 10th birthday. The cake said, "Happy 10th birthday, neuromorpho.org." 50,000 neurons and counting did not get to be written on. But a lot of the people, some of these people here you recognize because they're obviously here at Krasnow, others are alumni, including some from back in 2006. And what is interesting here is that, of course, as we keep adding data, the complexity of the structure of these data also grows. So this is along a few dimensions, the slide from Sridevi Palavaram. So at the beginning, the number of species in the database were maybe 10 or dozen, and now we're looking at almost 150 different species, and different species were added at different times. Same for experimental conditions and brain regions. So now the number of concepts that is expressed in the database to make that database searchable and so forth is in the many thousands. So what people do? What do people do with these data? So the first question is to identify the neurons. So the shape of the neurons is basically the main way that people recognize these neurons. So that's one of the reasons that they are collected. A big community usage is for computational modeling. So you basically have the explicit structure of the dendrites and the axon, and you can, with appropriate mathematical equation, predict or compute what the electric flow of information will be in those structures. All the delays, all the integration, the sublinearities or superlinearities, and so forth. There is a very rich, flourishing community on that. Another one is to actually compute quantitatively the differences among conditions, for example, so comparative analysis, if you like, and finding out if two groups of neurons are different over whatever dimensions, then what are the parameters that make them differences? Is it just size? Is it how flat or fractal they are? Is it the bifurcation angles and so forth? And all the data is there explicitly represented in these reconstruction files. The third one, which is becoming more and more popular for the reasons I explained, is to compute the potential connectivity. So you can take the dendrites of one neuron, the axons of another neurons, and sweep the space of possibilities to say what is their optimal placement to have a greater chance to form synapses, for example, and so forth. And finally, there is quite a bit of data mining, education outreach. There are now MOOCs that are created massively online, open courses that are using neuromorpho.org material in more and more cases. And so this is kind of a quick statistics of the downloads. We're talking millions of neurons downloaded for different species or different brain regions and so forth. And in terms of reuse, there are many articles, that peer-reviewed articles. So we're talking about academic throughput, output of people who have used data in neuromorpho.org to demonstrate or formulate hypotheses of new things. These are some of the earlier ones that came out. There are many more now. But the striking thing about these four that I set out here is that they all require data from many different conditions, many different brain regions, because they're about general principles of how dendrites or axons work. And so you could not really do this with a typical preparation from within one lab. You need the community to contribute data from many different studies to extract those. And lastly, because academia is not all, neuromorpho.org has been used in a variety of different settings. One of the earlier ones was the China Applied Math Olympics, where they used neuromorphology from this database to ask questions about applied math. We got about 100,000 downloads over a long weekend and nearly crashed our early servers. It was named Site of the Month in Neuroscience for Kids, which is a very popular website. It's popular among adults as well. It was showcased in Scientific American. It received a bunch of positive reviews all throughout both journals and books. There was a conference that was funded by Burroughs Wellcome here at Mason on data sharing. And perhaps one of the most consequential was that we were invited to deliver a full testimony to the Presidential White House Bioethics Commission as to why there is not more data sharing in neuroscience. And of course, they took the easy part, which is to ask the people who are being somewhat successful at data sharing. So my answer in basics for them was you should ask the people who are not being able to share right now. But still, the problem, even in morphology, it remains that more than half of the data is still not shared. So there is a lot of resistance in the community about making these data public, which is not there in other branches of, say, molecular biology or physics. And this is a slide.
When you talk about data sharing,
yes,
at any rate,
yeah,
that half the people are so
yeah,
aren't willing to do that. So articles such as author so and so publish a peer-reviewed article in
yes,
such and such, and are they, and let's say it's in one of these specialized areas that you're particularly focused, let's say hippocampus in elephants. Do
yes.
you then, having come across this, then inquire from the authors for their data? So what I'm trying to get at is, how do you,
yes,
only a part
yeah,
of
absolutely. No, we ask for the data, and in some cases I simply do not reply. In some cases I reply saying, nope, we're not going to give you the data. So in some cases say, oh, the postdoc has left, the computer was not backed up, and we don't have the data anymore. So this is, there's no need to say so and so. We actually have names, PubMed IDs, and so forth with reasons as to why the data were not there.
What's the statistic? How many reply yes? How many?
It's about half and half.
Half and half.
So when we actually publish some of these data, the reviewers call these two links the wall of fame and the wall of shame, because we make the data public. But this is still not, in a way, if it's their data, it's their choice. I think it's an unwise choice, but we can argue as to whether it's nice or not. But I was stunned because we have one of the former NSF program officers, who are a professor here at Mason, who gave a talk in my center last week. And he actually has statistics from the grant, from NSF, where in the grant, which is a legal federal document, people say we will share those data. And now they have the 10-year report, and half of those people are not sharing the data. I mean, to me, that's stunning, right? Because it's a contract in theory, right? So it's not just that you did it on your own and it's your data.
But when you say the data, so there's, let's say, an image of a dendritic arbor of a neuron in such and such a region.
Yeah.
You don't want just the
I don't want the image.
Itself.
I don't want the image at all. I just want the digital data. We only ask the data that we know were digitized, because we read the experimental methods and they say, we trace the data with Neurolucida or with via 3D, and we know that these softwares produce these XYZ. And I just want to finish with my acknowledgments, but before that, I had one slide that I had put out for our computational social science colleagues, which is the interesting meta-statistics that we did, and these are data from Patricia Maraver, who basically looked at all the IP addresses and where data are being downloaded and where the data are coming from based on the labs of people who are sharing the data, and then you can compute that ratio and relate that to the gross domestic product and see how that trend changes over time, and basically look at developing countries versus not. And it's actually very interesting. I'm seeing that more and more. I'm being invited in giving talks to countries that you would not necessarily associate with very prominent science, and yet they are emergent. They don't quite have the -80 freezers. They don't have the super-duper microscopes that we can build in the US and in Europe, but they certainly know their way about computer science. And for them, having the data online would produce an absolutely multiplicative potential for discovery. So I mean, I'm very passionate about the fact that if the data were all online, humankind would be, in fact, much better off, because we would basically get more brains at work on the interesting questions. So I'm going to close here, first by thanking the current grants that are supporting my lab from NIH, from NSF, including collaboration with Ken DeYoung, and with Dan Cox, who used to be at Krasnow and is now at Georgia State University. A new NSF grant that just arrived in collaboration with our affiliate, Jacopo Nese, and nonprofit such as Burroughs Wellcome, or corporations like Northrop Grumman. And the people who did a lot of the work, these are the current members of my lab, postdocs, specifically the people working on neuromorpho.org, are Ruben Ormananzas and Sridevi Palavaram, graduate students from many different programs: bioengineering, neuroscience, bioinformatics, and programmers and postbacks that are doing most of the coding and a lot of the new data extraction, and then a very valuable team of data processors on whom we rely to annotate these data and to correct these data because there is tremendous human-based quality control. Somebody has to go through these neurons one by one. So if there are 50,000 out there in the corridor, somebody inspected them one by one and made sure there were no mistakes. And even if the error rate is maybe a few percent, which is pretty typical in biology, that means on a data set of maybe 1,000 neurons, you still have maybe 30 or 40 neurons to correct in that many days or weeks of work. And finally, if you go to our website, we list all of our former collaborators and alumni. We have now several hundreds of them at all levels, and we try to stay in touch with them. We're very proud of where they ended up. Several of the former neuromorpho.org teams from the last few years are now at Janelia, working on similar endeavors on that end, and several others in many other academic venues. So I'll stop here. More questions, I should say, since we had some discussion over the talk already.
So what functionality is missing and critical? So you could wave your magic wand and say, what next 10 years? What do you need?
I can tell you right away. I think that there is one very difficult issue, which is linked to what I'm personally interested in, which is I would like to have the embedding of these neurons in the real tissue. So right now, the coordinates are arbitrary. Right? I have to literally take these neurons. I know that this neuron is from the retina. I know this neuron is from the cerebellum, but I don't know where it is. I don't know what the orientation is relative to the tissue. Right? And part of the reason is that those data were not out when the neurons were collected. Now we are in a place where there are atlases, so people are moving towards extracting the data directly registered into a common coordinate framework. And I think that will make a huge difference.
So there is now a common coordinate framework which is useful across.
It was literally announced last week by the Allen Institute, and it's on Twitter right now. I don't think they have a peer-reviewed outcome of it yet, but I was in the teleconferences coordinating the various resources to make sure that when it's rolled out, places like neuromorpho.org will be able to receive. And this is, if the data come, it's not difficult to put it into the structure. I mean, we know how to do it. The question is people have to collect those data in a way that contains that information.
I'm not sure I quite understand that. So are you saying that when you say you'd like to see the histological section, so do you want, I'm not sure.
No, I don't want to see the histological section. I want the XYZ coordinates of these branching structures to mean a particular location in a standardized brain of the mouse or the fly or the worm, depending on where those neurons came from. Okay? So that then I can take, say, the 10,000 neurons from the rat, I can put them in the proper location in the atlas and zoom in and look at their statistical, because they're coming from different animals, but statistical relationships as to how they might interact with each other.
So to say it another way, is you want the barcode address of where the neuron is in a brain.
Yes. Address and orientation. It's not just a location, you need two coordinates, right? But all you need is two 3D coordinates. Because if you have 2 points, it's 3D, you're defining the whole thing. Yes.
So you're talking about the latitude and longitude of the brain?
That's right.
There hasn't been something like that?
Big cartography. No? Incredible.
There's so much diversity in one structure.
That's right.
Yeah, that's been the issue.
That's right. So part of it is how to, in fact, match it to a common coordinate. And the reason that the Allen is coming out with that, by the way, in the mouse only, right, it is because they literally went through 300,000 mice, and they actually have the images of all of those brains. And what they found out, instead of doing very complex computational geometry, is that if they simply average all the mouse brains, all the noise goes away, and you get a fantastically perfect standardized atlas. And the nice thing about it is that you know exactly transformation, because you basically have a standard deviation of each one. So you're averaging, so you can get the second and the third moments and so forth. But again, this is something that became possible because it was one place doing the experiments in always the same condition, the mouse of the same age, the same breed, the same microscope, the same thickness, the same stain, and so forth. Right? So typically, this is not how it's done. Yes?
I have a different request for neuromorpho.
Yeah.
I don't think you have, which is we've been unable to use any of those neurons because there's no diameter.
This is a great question. I don't know if you know that is actually annotated. It's not true that there is no diameter. Some neurons have diameter, some do not.
I'm interested in.
Yes. So if you go, so then you have to ask the experimentalists to collect this data. But if you look at completeness and you look at the morphological attributes, you can see here whether the diameter is there or not. And so basically what you have to do is search for that information as well as the brain region, the species, and the neuron type you might be interested in. Another thing that I think is very important is that I've been talking about neurons all along, but glia are also tree-shaped. And we're now adding glia to the database. More and more labs are constructing glia. And so ideally, we'll be able to put it all together to create little tissues. The biology more biological. Yes?
So what do you think about using this as like a validation mechanism? So of course, there are going to be beginner people who are looking at morphology and who want to be sure that the numbers that they're getting are valid.
Awesome.
So how could they use this?
This is an awesome question. And let me tell you, we have a paper that is under review in Nature Methods right now. And the reviewers, in fact, asked us, why don't you just distribute it to the community? Why don't you let people do their own checks and so forth? And our answer is, well, if more than half the people are not willing to give us the data, if we do all the work, you can imagine if we make them do the work. However, the strategy we're devising is to progressively share the tools that we use to do the quality control, for example, try to share them in a user-friendly way. So there will be a website, maybe connected to neuromorpho.org. And if you're collecting your own data, you can say, hey, is there any mistake in this data? It's not just visualizing, right? Because you could look at something that looks perfectly nice, and then inside there is a 0 diameter point, or there is a dendrite that goes up and goes back down 3 or 4 times before it branches. And those things are very common issues that we find. And so the idea is that you basically go to this website, upload your data, and immediately, if possible, it would actually self-correct. If not, it would color code, say, hey, you have some issues here, here, and there that you might want to edit. And that's one way to do it. There is another one, which is in the search for morphometry. This is already out there. You can search data for morphometry, and people basically don't know what the ranges are. Wi-Fi might be jamming here. Never mind. So if you search by morphometry, you can, oh, there we go. You can search, for example, say, I want to search neurons that have a certain number of bifurcations in their trees, but you don't know what that reasonable number is. Is that 30? Is that 1,000? Is that, what is it? So you can click here, show sample, and it will sample randomly the database and give you basically a distribution of quartiles. I'll say this is the minimum, the maximum we have. So in this case, most neurons in the database have of the order of dozens of bifurcations, but you have some neurons that literally have thousands. So you can basically get, now, of course, this means that this is all across species, so it's quite possible that the sensory antennas of the moth that have spectacular bifurcations, they're supposed to be sensitive to micropascals of pressure in the wing detection, probably have the most, and maybe standard neurons that have just regular tree shapes might have only 50, say, bifurcations. But that's another way that you can explore. Time for trick-or-treating? All right, happy Halloween.